Thank you all for joining today. My name's Bill. I'm the Director of User Services at the University of Chicago. And today I'd like to present to you uncovering patterns in genomic variation data with clinical phenotypes using the new GDC Correlation Plot Tool. I'm here with my colleague Shin. I'll let him introduce himself at his section, but we will get started. So just to provide a few details, Sharon just went over them, but if anyone just joined, I also want to mention them. First of all, the webinar will be recorded. The recording and the slides will be made available soon. We need to do some processing. We need to add some transcripts. So it takes a little time, but they will be made available on the GDC website. And if you have any questions, actually type them into the chat panel. The Q&A panel must have been something different happened with WebEx since we made these slides. So we will address any questions at the end. So feel free to type them in as the webinar goes on, and yeah, we'll answer them at the end. So just to go over the agenda real quick, I'm first going to give an introduction to the GDC data, which is what the correlation plot tool uses. Then Shin's going to talk about the correlation plot tool and give a correlation plot tool demo. Following that, if we have some time, I may give some more information about some other GDC resources, but mostly we'll be doing a Q&A session. So any questions you put into the chat, we will address then. So to start, let's give an introduction to GDC data. And I first want to mention that there are two main data types available in the correlation tool. The first is the directly submitted metadata, which are the case properties and the clinical properties. These are metadata that are submitted directly by the people who provide the data to the GDC, the submitters. The molecular data generated by the GDC is the other type, and this includes somatic mutations, copy number estimates, and gene expression data. I'm going to get into both of these in the next slides. So to talk about directly submitted metadata, some types of GDC data are uploaded directly by external submitters, and this adheres to the GDC's data model. So some of you may be familiar with the GDC's data model, the data dictionary. All that data adheres to that. So all the data you see will adhere to that. And this includes case data. This is information that's directly about cancer patients and cell models in projects that involve cell models instead of patients. And this includes things like the primary site where the primary tumor was found anatomically, the disease type, which is a larger scale classification of the disease, something like adenocarcinomas might be one. And loss to follow-up is another example of like information about the case specifically. The clinical data includes phenotypic data about a case that was collected in a clinical setting, usually during a doctor's visit or a hospital visit. This includes information about the diagnosis, like direct information about what was used to diagnose the cancer, what the exact diagnosis was, demographic, information about the patient's demographic, like ethnicity, race, things like that, and then family history, information about family members that also may have had cancer that are related to the case. So I didn't include this in the first slide because this is kind of like almost halfway directly submitted metadata and halfway GDC generated data. And this is survival data. It was derived from several clinical time point values. So this includes, at point 0, we have the date of diagnosis, and then when the line decreases, that comes from either the days to death or the days to last follow-up. So these are the specific numbers that the GDC pulls from the directly submitted data, and then that data is these curves are generated dynamically using that. So next, let's talk about the GDC generated data. So the data that's not pulled directly from submitters or provided to us directly by submitters is data that was derived from molecular data and generated using the GDC's pipelines. So first I want to talk to you about somatic mutations. These are SNV data. or sometimes small indels that were generated from somatic variant callers. In the GDC, you might hear it called SSM for simple somatic mutation, and that just includes SNVs and small indels. Raw somatic variants, so that would be usually in VCF format. Those are annotated with information about the consequence, which gene it affects, and then they're conservatively filtered to remove the chance of germline leakage. So at all of these steps, the files are available, but the information that is used in the correlation plot is the end result of this pipeline. So this is variants that have no chance of containing any germline. So this is all somatic variants, and it's open access. This is data. If you want to access this through the API, this is available through the SSM occurrences, and there's some more endpoints. But if you're looking for the files that these come from, they're the masked somatic mutation map files. And an example that you might see in the correlation plot would be an SNV or an indel in SSM in the gene TP53. The second type I'd like to talk to you about is the copy number estimate data. This is a classification based on copy number estimate integer. And we classify this as amplification as the highest one, followed by gains, wild type being no additional copy number or lower copy number, heterozygous deletions, and then homozygous deletions. So that's the range of their from the most copies to the least number of copies. This data is based on several experimental strategies and some workflows that include the workflows are ASCAS NGS, and that comes from the whole genome sequencing experimental strategy. ASCAT3, that data is processed using the SNP6 array data. And then we also have absolute leftover data from also SNP6 array data. I know the other data will eventually be available, but for now, this is what's there. And a correlation plot example would be a CNV gain in KRAS. And then the last data type I'd like to talk to you about is gene expression. This is generated from star alignment followed by star counts and FPKM-UQ. So this is performed using some custom scripts from the GDC. So taking the star counts in as raw data and outputting the FPKM upper quartile data. So the values may be binned or continuous. And if you're looking at the API, this is available through the gene expression values endpoint. And if you're interested in files, this originates from the gene expression quantification RNA-seq files. So this is where you would find them in the repository. So a correlation plot example using the binning type would be the FPKM UQ of TP53 from 15 to below 20. Just to provide a little more information, everything that I showed you is a very small subset of the data available at the GDC, and this is specifically the things that can be analyzed with the GDC Correlation Plot Tool. So there are lots of other data types available from the GDC, and you can download them from the repository. There's some other API endpoints, but for more information, we have a documentation site. There's a repository, and if we have time, we might show you some of those at the end. I also want to note that there are other webinars about data types, so if you're interested, go look those up. So now I will pass the presentation to Shin, who will give a demo on the correlation plot tool.
Thank you so much, Bill. Hi, everyone. I will please give me a few seconds to share my screen. I should be sharing my screen now, in which I display the GDC data portal. I hope you can see it. I will demo. Right now, I'm not logged in, so all the data I show is open access. To access the correlation plot, you can find it under the analysis center. and under the analysis tools section, you should look for this card called correlation plot. So here I will click on the play button from the card to launch this interface. This correlation, as Bill has introduced, the correlation plot allows you to correlate mostly the 5 data types available from GDC, which are clinical, survival, somatic coding mutation, gene level copy number variation, and gene expression. All of these data types are accessible will be accessible from a unified interface from the correlation plot. Here, as an example, here you can click on one of these blue buttons to show this interface. At the right side of this panel, it will display the survival variable as well as other clinical variable here. The mutation and CNV are accessible via this second tab. The gene expression is accessible from this third tab, which I will get to it later. And I want to mention this interface corresponds to the correlation input button over here on the top, which is a generalized way to choose variables for comparison. Next to this correlation input button, there are additional buttons with different names. These buttons are designed to provide quick access to frequently used comparisons. I will begin by begin with the first quick access button called mutation CNV versus disease type. So now I will click on this button and it will show this input menu here. I will just launch a quick example by typing in IDH1 at the gene search box. While the chart is loading, this correlation analysis is limited to cases with mutation data because the IDH1 mutation or the mutation for any other gene is used. So the total number of cases showing here in this chart represent the cases with mutation data here. And cases without mutation, with mutation status unknown, or they do not have DNA sequencing, will not be represented. So this is a result from the mutation CNV versus disease type by comparing the IDH1 mutation status with the disease type. This is a result showing as a bar chart. Each row is one disease type, as you can see, and the stacked bars are IDH1 mutation status, with pink for IDH1 mutated and gray for IDH1 wild type. This is also shown at the legend down here. Again, the number of cases per disease is restricted to cases with known mutation status. All right. So I should also mention that this demo as well as all my subsequent demo are based on this cohort setup from the GDC portal, which is using brain or breast from the primary site. This is to limit the number of cases so that these plots can run faster. If you use a bigger cohort or not to use any cohort at all, this plot right now will run slower. So back to the bar chart. It shows, we can see that it shows the subset of gliomas, disease type cases have IDH1 mutation, as indicated by this pink block. And I hover my cursor over this pink block. I see that from the tooltip, it is 38.1% with 435 cases with IDH1 mutation. And subsequently, it is 61% with IDH1 wild type status. And next, I can click on either of the block to access additional option. I click on this pink bar to access these options here. I will next click on list cases, which will take some time, and will list all the cases from gliomas with IDH1 mutation. So here, this is the result. The case submitter ID is shown on the first column with a hyperlink. You can click on the link, which will take you to the case page in the GDC portal. So in addition to that, the actual IDH1 mutation is shown for every case, including the amino acid change and the consequence. It seems like most of these are silent mutation, are missense mutation, and with the R132H as the hotspot. There are a few different other kinds of mutations. I will next go through a few additional ways to utilize this mutation CNV versus disease type, which allows this particular tool to be customized. These customizations are also can also be a commonality between the other type of correlation tools. This first of all, this tool is named by disease type, but it's actually not limited to only correlating genomic alteration with disease type, with a particular disease type variable. What's shown here for disease type over here is actually a interactive button. I can click on it and then access this replace option. From this menu, I can select a different clinical variable to be correlated against mutation CNV. As an example, I will select a primary site. So now primary site is shown instead of disease type. I will still use IDH1. Let's wait for the plot to show up. As you can see from the hooded, now it shows. Now instead of compared to the prior plot showing the breakdown by disease type, now this new chart shows the breakdown by primary sites with breast and brain, which correspond to the cohort filter I set up. And we can still see that most of the IDH1 mutation are concentrated in the brain primary site. And also, you can see the correlation plot can keep multiple plots open at the same time. This is an intentional design to make it easy to cross-reference related information from multiple plots without having to flip any kind of a tab or chase results. There are additional ways to customize the mutation CNV versus disease type. So I go back here. I will next talk about the multi-gene mutation. What I demonstrated is a single gene mutation. In order to use to assess mutation status from more than one gene, I will add this input type radio button selection here. I will change it to gene set. Now this different input panel shows. As an example, I will enter, let me see. I will enter 3 different genes. IDH1 as what we already used, ATRX and P10. These are all genes with frequent recurrent mutation in brain tumor. So I will just next hit enter, and all these 3 genes are validated and shown in this box with the submit button highlighted, ready to go. So I just submit. I want to mention before the plot loads up, I want to mention this feature right now. This gene set mutation feature right now is limited to up to 10 genes due to performance issue. Once we further improve its performance, this limit should be relaxed so that you will be able to utilize more gene sets such as those available from the MSigDB, which are much larger, which usually the size of gene sets are much larger than 10. Now, this bar chart now shows this compared to the single gene IDH1. mutation status in the previous bar chart. Now we see the pink portion from glioma has a substantial increase. By hovering over here, we see 60%, nearly 61% from glioma has a mutation from these 3 genes. This is for a glioma case with a mutation in either of these 3 genes will be counted into this pink group. So we can see the details by clicking on this pink, and I select the load list cases. We can see the detailed mutation status for them. Now it shows. So it's a similar looking table shows. So now it shows the actual gene as well as its corresponding mutation. You can see that some of the cases actually have most of the cases seem to have a single gene mutation from have a single mutation from one of these 3 genes, such as all of these. And there is this particular case with both ATRX and the PTEN mutation at the, in the same sample, right? So I will move on to talk about the correlating copy number status with disease type or different clinical variables. In order to do that, I will go back to this input interface and select a CNV option here. To demonstrate CNV, I will use a different gene called EGFR. Now let's wait for the plot to load. So this plot now shows the EGFR copy number, gene level copy number status overlaid on the different disease type, where the different CNV categories are shown in the legend over here, which was also mentioned in Bill's introduction, where the bright red represent the amplification, and we see that it is concentrated in the gliomas, which is correspond to known cancer biology. So this concludes this first part. I will next move on to talk about the mutations, CNV versus survival. By clicking on this, it shows exactly the same part of input, the top portion of the input, exactly the same as the previous example, mutation CNV versus disease type, except now it's comparing against survival instead of any a clinical variable. So I will use the same IDH1 example, IDH1 mutation, and compare that with survival. It should be comparing the IDH1 mutated versus IDH1 wild type. So this Kaplan-Meier, this survival plot, also known as a Kaplan-Meier plot, now shows great differences for it displayed 2 curves. The 2 different curves, the meaning of the 2 different curves are explained at the underneath at the bottom of the survival plot. In the at-risk portion, the gray is for wild type, and the pink red is for IDH1 mutated. And we do see that we do see it's a striking difference in that the IDH1 wild type actually has a worse survival compared to IDH1 mutated. We can I can do this survival comparison in different organ types. I will do that by finding this menu button. Click on it. It will open up a configuration menu here. I will find this divide by variable with a prompt. I'll click on it and select primary site. Now I'll wait for the survival plot to be carried out in different primary sites. So I will hide the menu in order to see it better. So now the survival plot is showing results for is now showing 2 separate survival plots. With the top one is for cases from breast, and the bottom one is for cases from brain. Now we see the most striking difference between IDH1 wild type and IDH1 mutated survival differences is observed from brain, and this corresponds to known knowledge from cancer research. So I will go back to the mutation CNV. I will next talk about demo the CNV assessing correlating gene CNV status with survival. I'll go back to I'll click on this CNV versus survival button and check the CNV radio button. I will use EGFR so that I can assess EGFR CNV status with survival. Let's wait for the plot to show up. Now the plot shows which with multiple curves. Each curve represent cases with different one type of EGFR copy number status. With again, like what we see in the CNV bar chart, the red curve represent the cases with EGFR CNV amplification, and accordingly, concordant with the EGFR oncogene status, this cases with EGFR amplification show the worst survival. So now I will go back to the navigation button here. I will move on to demonstrate this CNV versus mutation. So here I will demo P10. I will use gene P10 to demonstrate that. Let's wait for the CNV and mutation comparison for P10 to show up, which now show up as a bar plot. The different bars, represent the mutation. So both the bars as well as overlay, they are all from P10, from the gene P10. The bars are the mutation status. The overlay are copy number status. What I would like to point out, and then we will divide this chart by primary set. So that we can see the same P10 mutation versus CNV comparison between two different organ types. This is similar to what we derived from the survival plot. Now we have two different bar plots, one for brain, one for breast. We can see that I would like to point your attention to this green block, which represent heterozygous deletion for the P10, and this also, actually in this mutated category. This suggests double hit because this is in these samples, P10, one copy of P10 is deleted, but also these samples also harbor P10 mutation here. So this suggests double hit, and these double hit cases are mostly from brain. And certainly I can, like what I did before, I can click on any of these blocks, such as this block I suspect to have a double hit, and list these cases to see details, which shows every item, every row is one case with their P10 mutation, actual mutation, and then the copy number status, which is all heterozygous deletion. You can browse this list. I will next move on to talk about the CNV versus gene expression. By clicking this button, it will show this menu. So I will use EGFR to demonstrate. Now let's wait for the EGFR gene expression to show. This is a violin plot showing the EGFR with numerical axis for UQ-FECAM values as a measurement for gene expression in GDC for EGFR, and the violin plot shows the distribution of UQ and FECAM value in different groups. These sample groups are divided by the EGFR copy number status, and we can see that the copy number cases with EGFR amplification has a significantly higher EGFR expression compared to the rest. I will talk to next move on to talk about the last quick access example, which is gene expression versus survival. So again, I will use EGFR. Let's wait for the survival plot to show, which will be dividing the cases by EGFR expression cutoff. Now the survival plot shows three curves representing three group of cases, GDC cases, and from the at risk legend down here, we see by default it uses a particular cutoff value, UQ-FPN cutoff value of 6.82, which should be the median value for EGFR expression. The blue curve represent cases with expression under this cutoff. Orange represent cases with expression above this cutoff, so lower expressed and higher expressed. And there's a third group with missing data, which doesn't have any gene expression data. I would like to mention the EGFR expression in when you compare gene expression with survival. This cutoff value with gene expression cutoff value can be customized. In order to customize that, you can click on this config menu button to access the configuration options. So at the top, it shows EGFR UQ-FPN with indicator of 2 bins. I will click on this tag and access is edit. Select the edit option. So this will take a little bit of time to load up the customization interface. This new interface allowing me to customize the setup of the EGFR gene expression variable or the binning criteria for the EGFR gene expression. So we see that without going through all the design detail here. So you see from this text box, it has this pre-calculated 6.82 as an example here. So I will choose my own setup cutoff value, which is 10. I will hit enter, so I can enter the next value, which is 100. I hit enter again, and the UI user interface will revise because I use 2 different cutoff values. It will allow to divide the cases into 3 groups: lower than 10, 10 to 100, and above 100. So high expression. I will click on apply. And wait for now. We actually see that behind the cloak, the expression variable already updated to show 3 bins, which correspond to the how we set up that. And on the survival, it shows 4 curve, which include the missing data part. And what we see that here is, if I zoom in, and you can see blue represent the low expression, and orange is the medium, and green is the high expression part. And so we see this gradients. The higher the EGFR expression, basically pretty much the worse the outcome it is. So now I have finished talking about all these quick access buttons, these 5 of those. So I will now circle back to the initial, this correlation input. I will click on that. It will create this input panel into my vertical carousel of all my plots so far. So I will scroll back to this input panel here. So you can use this to replicate. You can totally use this input, correlation input to replicate any of these 5 quick access already there. But also you can use it to set up comparison scenario which are not covered by these quick access examples. As example, I'll give you a few quick example. As a primary variable, I'll click on it, and from the clinical dictionary, I will select a disease type. And the variable to correlate, I will click on it, and let me see, I'll go to demographics and select a sex at birth. And now I will press the submit button. Let's wait for the sex compared to disease type bar chart to show up, which now shows up. So as shown, like now the 2 different, now the overlay has 2 different colors which represent the majority of these are from for male or female from the sex at birth clinical variable. There's a minority with not reported, and we can see that some of these disease type has near equal proportion of male and females, and some other like are dominated by female, which is totally makes sense given their organ of origin. So this is an example of correlating two dictionary variables. So I will go back to create a second example. At the primary variable, I will select survival. And at the variable to correlate, I will select primary set. Let's wait for this new survival chart to show. So this new survival plot shows the survival comparison by breast and brain using a clinical variable. Okay, I think that this concludes my demonstration of the correlation plot. Thank you very much. I'll be happy to answer a question at the end.
Thank you, Shin. And I don't see any questions yet, but while we wait for some, let me show everyone around some of the other features in the GDC. So I'll go ahead and share my screen. And I put up this slide, but I would rather just show people what's going on. So I'm going to open up Google Chrome here, and some of these websites you might hear, docs.gdc.cancer.gov, portal.gdc.cancer.gov / whatever. You can get to all of them just by remembering gdc.cancer.gov. So all the sites link off of this one. But there's a few things I want to show you first. So one is if you want to look for the recording of this webinar or any other past webinars, you can click on support, followed by GDC webinars. And all of the webinars we've done in the past are here. They all have associated YouTube videos, and most of the ones over the past few years do have the slides along with them. So you can download them by clicking on the specific one and clicking on the maybe that one didn't. Oh, here we go. The presentation down here. So if you want to get to the data portal, you can click on Launch Data Portal, which will take you to portal.gdc.cancer.gov. You can dismiss some of these banners here to make more room. And to get to the tool that Shin showed, if you click on Cohort Builder, you can build your cohort, and then click on Analysis Center, and you'll see the card for it with the they're in alphabetical order, so you'll see it right about here with this logo. If the screen is smaller, they rearrange themselves, so I can't guarantee which row they'll be in, but that's where it is. And you can click on demo, or you can use it on your cohort that you're using. One more thing I'd like to show, and I'll use the link instead of going to the actual URL. So under GDC apps, you can click on this and all the different GDC sites are there, but documentation is where all of the users guides for all of these different tools are, including the one you just saw. So if you were to click on Data Portal, all of the different tools that are in the Analysis Center, all of these have their own dedicated documentation page here. So you would click on Correlation Plot, and this has an entire user's guide that reiterates a lot of the things that Shin said and gives you general information on how to use it. This applies for any of the other portal features, the API, data submission if you're a submitter, or information about things like how the data is processed. So I think that's all I have to show. So if there are any questions, we'll be happy to take those. Okay, I don't see any questions yet. So we'll give it just a minute or so, and then we will conclude if there aren't any questions coming down the line. And just to remind everyone, the Q&A panel doesn't seem to exist anymore due to some WebEx updates. So type any questions you have into the chat panel.
And if you have any questions later, you could certainly contact our GDC support.
Yes, and that can be found at, I'll go to the page. If you click on support, you can see support at nci-gdc.datacommons.io. Okay, so we just got a question. Is GDC like TCGA and CBioPortal? And so yes and no. So GDC does host TCGA data. So the most up-to-date TCGA data can be found in GDC. So if you were to go to the portal, and if you click on repository, sorry, not repository anymore. I've been here for a long time. If you click on cohort builder and you were to click on TCGA, you'll see that we have 11,428 cases from these. And this has all of the data that was previously in other portals that was related to TCGA, except it's all harmonized to GDC's data GDC's reference genome and all of GDC's bioinformatics pipelines. So some of the analyses might be a little bit different, but all the data is there. And CBioPortal and GDC are similar in that we both host analysis tools and things like that. I know there is some overlap in the analysis tools, but there are some that are specific to CBioPortal and some that are specific to GDC. So so yeah, there are some similarities and some differences, of course.
Yeah, some of the primary differences are like the GDC, we harmonize all the data using our standard pipelines. And so you can compare across projects. And our system is also cohort focused. So you can build a cohort, that goes across studies because the data is harmonized. And then you analyze the data associated with that cohort. That's just some of the differences.
All right, so the next question, is de-identified data available to international medical students for research? And the answer to that is yes. Everything that I'm showing you right now and everything that Shin showed you is available as open access. So if neither of us logged in, everything that I'm doing now can be done just by going to portal.gdc.cancer.gov. We do host files that are controlled access. So whether or not you could have access to that depends on an application to dbGaP, who are the ones who determine whether or not a project can have controlled access. But if you were to go down to this left panel here and click on access and you just click on open, we do have lots of platforms, lots of different workflows, and lots of different data types for that are completely open access. So I could just download these to my computer right now directly from the portal, or using a the data transfer tool or the analysis tools also. So in summary, yes, as long as in your country you can get to the GDC, you can access the open data. Whether the control data is available um, depends on your application to dbGaP. Okay, we'll give it just a minute for any other questions.
Bill, maybe you want to show them where you can get to information about our harmonization workflows, which is the backend data that drives the correlation plot tool that Shin demonstrated today.
Oh, for sure. So if you go to the documentation site, so docs.gdc.cancer.gov, we'll just go to the home page right here. If you're interested in how the data is processed, you would of course click on the data button right here. Click here. And then depending on what information you're looking for, you can click on, like, for example, DNA-seq analysis. So this is whole genome variant calling. This has information about that and some information about the actual pipelines that are run. So if you're interested in those, that is where you can find them. Things also like whole exome and targeted sequencing is available here. We have a lot of information about that because this contains the information about the alignment. And then also, if you're interested in the changes, because GDC deploys new data on a regular basis. So if you're interested in what the new data is, and what the changes are to the GDC, you can click on the release notes depending on which section you go to. So under the data release notes has the information about what new things are in data. So we're currently in data release 45. Here's the new projects and the new data sets that were released then. If you're interested in new releases from the data portal, we actually just had one a few days ago. And if you were to click on release notes, you'd see, yep, April 27th, version 2.7.0. And yeah, a new image viewer tool has just been released. So that's where that information can be found. And then if you're interested in things that involve the GDC data dictionary or data model, we do have this nice viewer tool that shows you which fields are available, for which type of entity, which type of data. So like demographic, for example, has all these different fields and these acceptable values. Um, so my apologies, WebEx is a little different than I remember it, but I don't see any new questions. So Shin or Sharon or anyone else, does anyone else have anything else to say? Okay.
Just thanks everybody for attending today's webinar, and we'll try to get the video and presentation posted as soon as we possibly can. Thanks so much, everybody. Have a great day.
Thanks, everyone. See you later.
Thank you.